Hello and welcome to the T-Level Science Content Buster, navigating the lab landscape to empower T-Level Science students. Please remember that the information provided here is based on data available as of November 23 and may not reflect future changes to guidance documents. Always refer to the most up-to-date guidance. In this comprehensive journey, we will explore the key components that form the backbone of effective laboratory practices. Real-world applications will be explored and understood and how to apply the content to real-world application. Through clinical skills ecosystem, we will explore the connection between all key themes today in relation to clinical skills. Throughout this recording, clinical skills linked to clinical settings, but as good practice, also have useful applications within general scientific settings. We'll also recognise the importance of adherence to quality standards, regulatory controls and health and safety protocols. We'll increase understanding the significance of standard operating procedures in maintaining standards and ensuring consistent, reliable results. And we will delve into the vital role of statistical tests in research, ensuring the robustness of scientific findings. This will equip you with the knowledge and skills needed to navigate the intricacies of laboratory science successfully. Now let's dive into the intricacies of standard operating procedures, or SOPs, which play a pivotal role in ensuring the smooth function of scientific and clinical settings. Think of SOPs as the guiding principles ensuring laboratory standards and consistency. They are the set of instructions that transform a series of tasks into a meticulously choreographed dance of precision. Responsibilities and compliance, SOPs outline the individual responsibilities with the broader team, emphasising adherence to organisation policies and procedures. Individuals have specific responsibilities within their team and the broader organisation. And adherence to SOPs contributes to organisational compliance with internal and external regulations, maintaining health and safety, ensuring consistency, meeting legal requirements, upholding professional standards, and demonstrating compliance for audit purposes. Individuals and organisations will be responsible for defining SOPs. Organisations enforce SOPs to ensure that all individuals follow the standard procedures, which tiers down to individual responsibilities around enforcing SOPs departmentally, for example. Individuals are required to follow SOPs, ensure that all SOPs are followed that are outlined for their task. Why is it crucial for everyone to follow SOPs? Consider these key aspects. Health and safety, detailed safety guidelines, hazardous material handling. Or through consistency, you have standardized procedures, uniform practices, minimizes variations and ensures reliability. Through legal requirements, it's important to meet legal requirements in regards to items such as safe storage of controlled materials. Uphold professional standards, adherence to codes and regulations, and it demonstrates compliance for audit purposes. SOPs aren't just documents, they guide us in working safely and efficiently in a laboratory. Following SOPs is essential when performing specific scientific techniques, ensuring compliance with regulations and maintaining quality standards. Applications in lab techniques, SOPs guide us in working safely and efficiently in laboratory. Following SOPs is essential for specific scientific techniques, ensuring compliance with regulations and maintaining quality standards. Through equipment and maintenance, SOPs go beyond techniques, guiding us in maintaining, cleaning, calibrating, and validating laboratory equipment. This includes interpreting manufacturer's instructions using the correct test equipment when following appropriate SOPs. SOPs play a crucial role in resolving issues with scientific equipment. They ensure equipment is working order, free from contamination, recalibrated per manufacturer's instructions and SOPs, and reset when necessary. As we unravel the layers of SOPs, we discover not just a set of procedures, but the backbone of safety, consistency, and compliance in our scientific endeavours. Stay with us as we continue our journey into the heart of laboratory mastery. As we step into the lab, where precision meets discovery, it's crucial to underscore the paramount importance of health and safety. Let's narrow our focus to the unique considerations within the laboratory setting. In the lab, strict adherence to regulatory controls is vital. This encompasses selecting the right personal protection equipment, or PPE, maintaining high health and safety standards, undergoing specific training, ensuring proper waste disposal, and meticulously following controls specified within SOPs. Within the lab, familiarise yourself with key legislation and regulation. The Health and Safety at Work Act, Management of Health and Safety at Work regulations are cornerstones, ensuring safety measures are tailored to the unique challenges of scientific experimentation. Lab health and safety promotion involves not only following protocols, but also modelling good practices. From meticulous hands-on practices to keeping workspace organised and hazard-free, every action contributes to a safer laboratory environment. Dealing with situations in a lab context requires following relevant SOP. Whether it's a spillage or an equipment fault, swift responses, securement of areas and thorough reporting are imperative. Within the lab, understanding and implementing REACH guidelines are paramount. These guidelines ensure the safe use of chemicals, promoting alternative assessment methods, and prioritising human health and environmental protection. The Environmental Protection Act 1990 finds a specific application in lab practices, particularly in waste management, containment, and the use of genetically modified organisms. Breaches have direct implications for the lab environment. Biohazards and cost regulations take centre stage in the lab. Understanding biohazard categories and strictly adhering to cost regulations are essential to mitigate risks to health, prevent disease spread, and protect the lab environment. In conclusion, as we navigate the intricate world of lab science, our commitment to health and safety forms the bedrock of groundbreaking discoveries. Moving on, let's explore the significance of statistical tests in research. Statistical tools are the compass guiding us through the vast sea of data gathered through lab work. They help us distinguish between mere coincidence and meaningful patterns, ensuring the robustness of our findings. Let's explore the various statistical techniques and considerations that contributes to robust analysis. To analyze data effectively, you must master several statistical techniques, such as mean and median, standard deviation for measuring dispersion, range for determining extremes, chi-square test for the significance of differences between observed and expected results, t-tests for group mean differences, and Spearman rank for assessing variable correlation. Selecting appropriate statistical techniques is crucial. Processing results using statistical software supports technical arguments, assesses statistical validity, and draws meaningful conclusions. Determining sample size is a crucial aspect, constrained by factors like cost and time. Sufficient sample size is essential for adequate statistical power, reducing the risk of errors in accepting or rejecting experimental hypotheses. Statistical power increases with effect size. When the effect size is small, a large sample size is needed to achieve a good power value. As effect size increases, the impact of a sample size on power is smaller. Precise calculations play a pivotal role. Take, for instance, the calculations of magnification or the determination of RF values in chromatography. These calculations aren't mere mathematical exercises, they are the bedrock of accurate data interpretation. Furthermore, the application of appropriate statistical technique is fundamental to determining the validity and significance of results, whether it's employing mean and standard deviation for result interpretation or conducting advanced statistical tests. Each technique is a tool for extracting meaningful insights. Let's look at an example. A researcher meticulously investigating the concentration blood protein levels using a t-test to compare the means of two experimental groups under the same conditions, ensuring statistical rigor in drawing conclusions. In both statistical analysis and lab environments, meticulous consideration and precise methodologies pave the way for scientific excellence. Let's now delve deeper into the nuances of statistical mastery within lab science. After conducting a statistical analysis, the next steps involve reviewing data against the original hypothesis, comparing with published results and checking tolerance levels. This critical review guides decisions on whether to collect more data, publish findings, or share results with clients. A possible thought process you could use to ensure effective data analysis could be beginning with review initiation. So you have a decision point. Is a review required? If yes, proceed to the next step. If no, end the process. Step 2, collect all relevant information for the review. So gather information. Step 3 is another decision point. Is the data reliable and complete? If yes, proceed to the next step. If no, end the process and highlight data issues. Moving on to step 4, statistical analysis, where you perform a statistical analysis based on the data. And in step 5, review statistical findings. You have another decision point. Do the statistical findings align with the hypothesis or objectives? If yes, you proceed to the next step. If no, consider re-evaluating the analysis or collecting additional data. Moving on to step 6, you interpret the results in context of the research objectives. And finally, step 7, decision point. So final decision, should you publish? If yes, proceed to the next step. If no, consider revisions or further analysis. Presenting data in an appropriate format is a skill. This involves using statistical techniques incorporating data from lab information management systems, or LIMS, for clear and effective communication with stakeholders. Contributions to scientific reports involve preparing concise abstracts, detailed introductions, comprehensive methods and results sections. Reliable and verifiable data presentation is key, along with identifying the most appropriate ways of displaying data. Let's explore these elements and connections in our clinical skills. Ecosystem. As mentioned previously, this applies to working in a clinical lab, but that many of the skills apply to general good practice. So at the heart of our system is the meticulous guidance provided by Standard Operating Procedure, or SOPs. These established protocols are the bedrock of ensuring consistency and safety in every lab task. Radiating from the SOPs is the domain of clinical skills, encompassing techniques like staining and microscopy. Adherence to SOPs is not just a recommendation, it is crucial for the successful application of these skills. Clinical skills connect seamlessly with statistical significance. Here, the precision of our clinical techniques contributes to reliable statistical outcomes. It is not just about performance tasks, it's about obtaining meaningful and statistical sound results. As we travel the landscape of clinical skills, we encounter health and safety implications. Using specific stains and microscopy techniques requires strict adherence to safety protocols. The integral connection between health and safety in clinical applications cannot be overstated. Enter the key player, the laboratory scientist. This entity embodies the culmination of SOPs, statistical analysis, and safety protocols in the lab setting. The laboratory scientist is the epicenter, ensuring that all elements harmonise in their work. However, every journey presents challenges. Addressing issues like technique-related concerns requires a keen understanding of the interplay between SOPs, statistical analysis, and safety protocols. Our laboratory science navigates these challenges adeptly, finding solutions that uphold the standards of the clinical skills ecosystem. In conclusion, our clinical ecosystem is an intricate dance where adherence to SOPs, statistical prowess, safety consideration, and the expertise of a lab scientist converge. Each element plays a crucial role, ensuring that the scientific journey is not just accurate, but also safe and meaningful. It is in this ecosystem that the magic of lab science unfolds, bringing precision and reliability to every analysis and experiment. So let's now look at some teaching activities to bring this content to light. to life. And in this example, we have the interactive workshops and simulations. The objective of those workshops would be students should be able to apply SOPs, conduct statistical analysis, and adhere to health and safety protocols, whilst effectively addressing challenges in a lab setting. The resources would be ensuring access to the necessary lab equipment and materials for hands-on activity, explore virtual lab platforms or simulation software that replicates lab equipment experiments, and provide comprehensive safety guidelines and protocols. And the procedure on the right-hand side, start with an introduction to lab equipment and their proper usage, design interactive experiments or activities where students follow SOPs, perform statistical analysis and adhere to safety protocols, facilitate group discussions to encourage collaboration and problem solving, and conclude the workshop with a debriefing session, allowing students to reflect on their experiments and challenges. And the teaching activity 2 is project-based learning. Through project-based learning, students will develop the skills to independently plan and execute lab projects, including the creation of SOPs, application of statistical analysis, adherence to health and safety protocol, and effective communication of results, showcasing a comprehensive understanding of lab mastery. And in the resources section, you would provide detailed project descriptions outlining the objective, expectations, and evaluation criteria, offer access to scientific literature, databases, and relevant research materials, introduce project management tools for collaboration and task tracking. And on the procedure, you could allow students to choose projects aligned with their interests with the realm of lab mastery, guide students through the planning phase, emphasising the creation of SOPs, statistical analysis plans, and safety protocols, and oversee the execution of projects, encouraging students to apply good scientific and clinical skills, conduct analyses, and address challenges. You would have each student present the findings, emphasising clear communication of results, adherence to SOPs, and considerations of health and safety protocols. Finally, finishing with incorporating a peer review component for constructive feedback and cross-learning. And our third activity, integration of case studies. On completion of the case study, students should demonstrate the ability to analyse and synthesise information from real-world lab scenarios, applying knowledge of SOPs, statistical significance, and health and safety implications to make informed decisions. So on the left, you would have the resources would be compile or develop case studies representing diverse lab scenarios, provide resources for students to explore real-world examples in scientific literature, offer guidelines for analysing and discussing case studies. And then the procedure, you would introduce the case study and its relevance to lab mastery component, divide students into groups to analyse and discuss the case study, addressing SOPs, statistical significance and health and safety implications, conduct a class-wide discussion to share insights, solutions and challenges, and assign a reflection papers for students to individually analyse the case study's impact on their understanding of lab mastery. Let's explore a real-world application scenario. So this is an example of a real-world application. This can be shared with students, which provides the scenario, so in this regard, the microscopic analysis of water samples, and the environment with the background and the environmental context. Here, you would also clearly see the lab's missions or purpose. And you would also share the microscopic targets. Followed by the regulatory framework the lab operates under, the expected community impact from the outcomes, and the case study objectives. So these are all the resources that you would begin with for your students. You would then give guidelines with headings as can be seen here. So your pre-analysis preparation microscopic analysis of water samples, you would task students with creating SOPs for the entire analysis process, include step-by-step protocols for slide preparation, staining techniques and microscopic examination, emphasize the need for clarity, precision and adherence to established lab protocols and SOP development. You would guide students in conducting a comprehensive risk assessment related to sample collection and initial handling. You identify potential hazards and risk associated with the collection process, emphasising the importance of mitigating risk through proper safety measures. Ensure that students calibrate and verify the calibration of lab equipment used in the analysis, such as microscopes and staining apparatus. Emphasise the significance of accurate measurements for reliable microscopic observations. You would step forward, discuss the importance of planning data collection strategies, including the selection of relevant microscopic fields, the identification of target microorganisms, and considerations for capturing images. You would introduce students to the basics of statistical analysis, emphasising the importance of planning for statistical tests during the microscopic analysis phase, discuss considerations such as selecting appropriate tests, determining sample sizes, and aligning statistical methods with research objectives. And finally, you would instruct students on proper procedures for collecting water samples, emphasising the importance of aseptic techniques to avoid contamination, provide guidelines for sample preservation, considering factors like temperature, storage containers, and the addition of preservatives. So that would be the pre-analysis preparation. For the post-analysis procedures, you would instruct students on the statistical analysis of their microscopic data, guiding them through techniques such as quantifying microorganism abundance, carrying out statistical analysis, and identifying statistical tests for significance. Emphasize the alignments of statistical findings with the initial hypothesis or research objectives. Guide students in interpreting their microscopic results in the context of water quality assessment. Encourage critical thinking about observed variations, correlations, or anomalies, and relate findings to potential environmental implications. Emphasize the importance of thorough reporting and documentation of the analysis process, including any deviation from the SOPs, unexpected results, and challenges encountered. Highlight the significance of clear and concise documentation for regulatory compliance and future reference. You would engage students in evaluating the effectiveness of health and safety protocols during the analysis, considering factors like PPE usage, containment measures, and overall lab hygiene. Encourage reflections on how safety measures contribute to the successful execution of the analysis. And finally, instruct students on effective ways to communicate their findings, both in written reports and oral presentations. Emphasise the importance of transparent communication with regulatory authorities, stakeholders, and the local community. So our key takeaways from today's session. We've understood how to apply the following content to real-world application. Exploring the connection between all key themes today in relation to clinical skills when working in a clinical setting. Recognise the critical role of quality standards, regulatory controls, and health and safety protocols in lab settings, and appreciate how standard operating procedures maintain health and safety, ensuring consistency and demonstrate compliance. And finally, understanding the significance of statistical tests in research, connecting data reliability to the core of scientific exploration. Thank you for watching.